Welcome back. Hope you all had a nice networking session. Let's kick off our technical sessions now. May I remind you all to close your cameras and mute your microphones, please. The first session is about biomedical technology, moderated by me. Biomedical technology, a multidisciplinary field that builds on understanding biological systems on the molecular level and develops new approaches to advance medical and biotechnological applications with an emphasis on human health and diseases. I am excited that we have eminent speakers in this session to share their research findings with us. Please use raise hand option to ask your questions at the end of each presentation. Our first speaker is Professor Oman Oman, Bioengineering and Nanomedicine Group, University of Tampere, Finland. Dr. Oman received his PhD in organic chemistry from the Indian Institute of Technology, Bombay, in 2010. He then moved to Uppsala University, Sweden, to pursue his postdoctoral research in the field of regenerative medicine. In September 2016, he joined Tampere University, where he established the Bioengineering and Nanomedicine Group, and he leads a multidisciplinary team. He works at the chemistry-biology interface, and his research involves biomaterials, nanomedicine, tissue, and cell engineering. He authored several peer-reviewed articles and book chapters. He has also filed 4 patents and is a co-founder of the spin-off called Uppsala Therapeutics AB that focuses on nanotherapeutics for brain delivery and nucleic acid delivery. Today, Professor Oman is going to present about glycosaminoglycans, a formidable biomaterial for nanomedicine, tissue engineering, and modulating immune response. Professor Oman, the floor is yours.
So today I'll be talking about polymers, the polymers that are produced by our own system. So we call them as glycosaminoglycans. These are polysaccharides that our cells produce. So we use them as a building block, and since they are produced by cells, we call them as extracellular matrix. So extracellular matrix polymers are the polymers produced by our cells. They can be proteins, carbohydrates, or a combination of both called proteoglycans. I don't know how many of you are from the material science or biology side, but I'll try to mix both together because I work on the interface. So my group basically uses the polymers produced by our cells and engineer materials. So we are basically using them as Lego bricks. If you think of a Lego brick, you can design any material, and you can choose polymers or proteins of your interest and design materials of different properties. From an engineering perspective, these are very interesting molecules. So I'm from a chemistry background or a chemical engineering background. So when I look at these polymers, I find a lot of functional groups. You can diversify them, you can do different chemistries on these substrates and can make materials of different kinds. But from a biological perspective, this is even more interesting. If you look at one example, hyaluronic acid or hyaluronan, which we have around 15 grams in our body right now, and is recycled very frequently in our body. And if you look at this polymer, these are the high molecular weight polymers technically have an anti-inflammatory behavior. They inhibit toll-like receptors activation, but upon injury, for example, if you have skin injury, the enzymes produced in our skin breaks these big polymers into smaller fragments. And these smaller fragments, on the other hand, are pro-inflammatory. So it's work as sensor. Can you imagine? Same polymer, no differences, just a difference in the molecular weight can make it a pro-inflammatory system or an anti-inflammatory system. So this is a magical molecule from a biological perspective, and from an engineering perspective, it's a very big toolbox because you can make whatever you want. By combining different properties of materials, you can tune the materials that you want. So in my group, we work on 2 different areas. One is making injectable hydrogels, and we also work on making nanocarriers or nanomaterials using these polymers. So my work confines to these 2 areas in principle, but we also do cell engineering, we also do stem cell transplantation therapies. So on the hydrogel side, we do tissue engineering, specifically bone engineering. We work with collaborators who work on cornea regeneration. We work with collaborators who work on cardiac regeneration. So these are 3 different tissues that we work on. By making materials specific to that particular organ, you can tune the responses, and you can recruit the stem cells from the surrounding, and you can reconstruct the damaged tissue. That is the principle of tissue engineering. And we also work on nanomaterials or nanomedicine for targeted drug applications. So in this short time, I will try to introduce two fields by giving some specific examples. These are some of the papers that are listed here that we published in the last few years. So first, I will talk about nanomedicine or nanotechnology for medical application. We recently published a paper. We have been making many different materials for nucleic acid delivery, specifically short interfering RNA and plasmid DNA delivery. We had been working on delivering small molecule drugs, but I will talk about some very recent findings that we made in our group. One of the studies we've made was on making a system that can deliver two drugs simultaneously in a synchronized manner for combination therapy, and where we succeeded in demonstrating that we can actually tune the release of the drug. Not only that, we also showed in this work that anti-inflammatory drugs can have a positive or a negative effect on a cancer therapy. And in traditional, we always give a cocktail of drugs where we give an anti-inflammatory drug as well as chemotherapy drug. So how they precisely regulate each other's responses was not properly studied. So in this work, we reported that, but this is not what is most exciting that I want to present today. Today, what I want to talk about is one work that we just published very recently, where we show that heparin nanoparticles can actually cross the blood-brain barrier and deliver drug to the brain tissues, and specifically the glioma if there's a brain cancer. So we made two different types of nanoparticles. One are called micelles, where we take these polymers and functionalize as hydrophobic molecules like fluorescein in this case, and that we can tune the degree of modification such that the size comes to the nano range. And we can also engineer gold particles by a very nice simple method where we can make gold in situ particles coated with these polymers. And we have taken two systems here. One is hyaluronic, one is heparin, which is known to have anti-inflammatory properties, known for anticoagulant characteristics, and many other things is used in clinic. And we also used a kind of surrogate for heparin called chondroitin sulfate, which is another sulfated glycosaminoglycan. We wanted to compare the two, and what we found, we first tested in vitro. So what we found was that you can see here, you have a bilayer system. You have endothelial cells on the one side, which represent the blood side of the brain, and you have and astrocytes on the other side, which represent the brain side. So the two sides of the brain we mimic in a vessel in vitro to make the artificial blood-brain barrier. And what we can see from the top image here, if I can use my cursor, no. Yeah, so from the top image, you can see that the heparin particles actually crosses over from the endothelial side to the astrocyte side. So this is very phenomenal because there are very few molecules that can actually cross BBB, and that is why we cannot treat many neurological disorders. There are many drugs available, but they cannot be taken over into the brain. So can we deliver something into the brain? It was really a very big question. So we actually accidentally found that heparin particles actually crossed the blood-brain barrier. On the other hand, the chondroitin sulfate, which is similar in structure, it's also sugars, it's also polymers, it's also sulfated, but it does not cross out the BBB. It is stuck on the endothelial side. Now, is it really true? So to that, we first did a very simple pilot experiment. What we did, we took a healthy animal with an intact blood-brain barrier, with no injury of any kind, no disease of any kind, and we did a tailwind administration. And what we could see, the fluorescent particles are visible in 1 hour inside the brain in these healthy animals. While on the other hand, the chondroitin sulfate, as we anticipated, did not cross over, did not accumulate inside the brain. And we screened different types of animals. We used the nude mice, we used the immunocompetent FVB mice and C57 mice to see if there's any variation between species, just an accidental. But we could see that in all of the cases, we could see it's accumulating in the brain, and it was not confined to one particular cell type. If you look at the bar at the bottom, we could see that most of them were just diffused inside the brain, and just around 10% into neurons, and maybe around 5 to 7% into astrocytes. So this is, microglia. So this is what we've observed. And now the question was, how much is going to the brain? How can we know what is the metabolism? How it is taken out? What is pharmacokinetics? There are many unknown questions arise from this experiment. So for that, we need something which we can image, we can quantify inside the system. So we made two different systems, one with a radio-labeled gallium, and other is gadolinium, which is MRI active contrast agent. So with these complex, with two different systems in these particles, and we first checked in a healthy rats. So we did an injection in a rat, and we can see in 1 minute, 0.34% of the injected dose reached the brain. If you look at this video here, if it works, it goes how it is translocated into the system. It reaches the brain very, very quickly, and it is cleared through bladder and is very, very efficient. Now, right now, we have done another experiment where we wanted to test if the tailwind administration is one option, but we wanted to see if we could do a lymphatic delivery. So we injected through a lymph nodes through a subcutaneous injection, and we could see very good efficiency as well. So that's something ongoing, which I'm not discussing right now. So likewise, when with MRI contrast system, what we found was very different from the healthy rats or healthy mice. In healthy rats and healthy mice, it just dispersed in the parenchyma. But when you have a disease model, when you have glioma in the brain, when you have tumor in the brain, it selectively accumulates in the brain tumor. The mechanism is still unknown. We still do not know why it crossed the BBB. We still do not know why it is reaching the glioma. We have done further experiments by delivering drugs to these molecules, and we could see that we can deliver drugs to the glioma cells inside the animals, and we could reduce tumor completely from these animals. But those are unpublished data, so I'm not discussing that here right now. Switching gears from nanoparticles, I go to hydrogels. So as I mentioned, we use these polymers to make hydrogels. Now, most of you have heard the two lectures in the beginning. They talk about hydrogels, very interesting talk about how nanocellulose gels are used for clinical application, for wound healing application, for 3D cell cultures. So we also make gels. We develop new chemistries, new methods of making gels, and we basically focus on cardiac and bone. These are two different areas that I work with. And we also engineer disease models, tumor microenvironment. We make gels for wound healing, releasing drugs specifically at a specific time, and we also develop bioinks. So most of you know about hydrogels, I believe, but hyaluronic acid hydrogel has a long history. I mean, they have been used in cosmetic industry a lot. You know, many of the ladies want to enhance the lips. The surgeon inject a gel, which is basically hyaluronic acid gel, that enhances the lips, and it stays for nearly a year. Something goes wrong, if the shape is not good, they can inject an enzyme that degrades these gels naturally, and you can remove it and do the surgery again. This is in use in cosmetic industry to enhance the features. It's used as in many anti-aging creams, as we know that these small heparin, hyaluronic acid, can cross the skin barriers and can actually rejuvenate the skin and in use for anti-aging creams. Also in arthritis patients, gels are injected in the knee so that regenerate the articular cartilages that is degenerated, and it is used in clinic. So there are a lot of clinical products on hyaluronic acid and hyaluronic acid based gels, both for cosmetic application as for clinical application. So we make gels. So one of the study that we just published this year, 2022, is that we wanted to make a gel that can be used for neurons. Neurons are very difficult to grow. They are cells which are very, very adamant to work with. They're difficult to work with. So we use this human iPSC-derived cells and embryonic stem cells-derived progenitor cells, and we cultured in our gels. The key question was, can we identify the matrix where these cells could grow? So we work with gels and ECM. One of the main components of the brain is hyaluronic acid and chondroitin sulfate. So we were having an idea, maybe hyaluronic acid chondroitin sulfate combination would be an ideal scaffold for culturing these cells. So we made some hyaluronic acid gels. I just showed some structures here. Don't get scared. Those were not chemists. Very simple molecule, very easy to work with. You can just functionalize with two different functional groups, which when mixed together, react with each other, cross-links and forms a gel. And what we wanted to test was, how do hyaluronan gels work in comparison to hyaluronan chondroitin sulfate gels? So we removed one polymer out from hyaluronic acid and replaced it with chondroitin sulfate, which is sulfated glycosaminoglycan, and we wanted to compare. And we also functionalized with dopamine molecules because they were also very useful for neuronal applications. So what we could see is a normal hyaluronic acid gel was not functional, did not do any good to the cells. When we use a combination of HA and CS, hyaluronic acid and chondroitin sulfate, we could see the sprouting of the cells, the maturation of the cells, but that was still not very efficient. In the literature, we saw that laminin mimetic peptides are very useful for these cells. So we functionalize these matrix with laminin peptides, mimetic peptides, but that's completely ruined the experiment. We did not see any effect at all. But what we found was, when we have used dopamine functionalized hyaluronic acid chondroitin sulfate matrix, the neuron starts to grow very fast. They had formed very nice network. There were long neurite outgrowth, and it was showing excellent results. And we wanted to investigate further why this was happening. And it was mainly because these matrices, when you have dopamine, dopamine is sticky. It sticks to proteins, sticks to substrates. So when you have dopamine in the matrix, it actually traps the ECM and remodel the matrix, and you can go more collagen production, more tubulin production, and MMP10, which is degrading the matrix, helping the neurons to sprout further, was much higher in this matrix that has both chondroitin sulfate and hyaluronic acid functionalized with dopamine. In another study, I have been very quick. I think I'm still on time. So we made another study very recently where we used another system. In the tissue engineering field, one of the big challenges is inflammation. When you inject anything in our body, the first thing that happens is inflammation. Inflammation is good. Rather, we need inflammation so that the immune cells would come there and start reconstructing the tissue. But the problem is, if the inflammation is prolonged, you get chronic inflammation, the whole system goes off, dies down. So many of the materials that we find, for example, chitosan gels or alginate gels or any other, even collagen gels, are inflammatory in nature. So you inject something, you have inflammation, and that causes problems. So we made a system. We wanted to do something which has anti-inflammatory properties. So when we look at different molecules, what we found was gallic acid is a small molecule which is found in green tea. It is found in many medicinal plants, and it is known to have strong antioxidant properties. We hypothesized maybe if we functionalize these matrices with gallic acid, we can make something which is more active in suppressing inflammation. So we made simple, same story what we did before. Instead of dopamine, we functionalized with gallic acid, normal HA gels, and what we found was something very fascinating from a mechanical perspective, that these gels were very stable, and we could grow them in cultures without any swelling or degradation. Just adding few percentage of gallic acid in the system, you can actually see on the bar on the right, you can see that even after 21 days, the gels are intact. But without the gallic acid, same hydrogel, in the absence of gallic acid, just swells and degrades within 3 weeks. And when we did a cell culture with immune cells, with macrophages, we could see that they were producing more immunosuppressive phenotypes, which was very interesting. And when we implanted in animals, we could see that was more myeloid cells accumulation in these gels, and they were mostly of immunosuppressive phenotype than what you generally encounter with other systems. So I just conclude very quickly that these polymers are very interesting polymers to make materials, both for drug delivery application and for tissue engineering application. You can tune the way you want, you can mix and match, you can, like a Lego bricks, you can design what property you want. You want pro-inflammatory, you want to last for a few weeks or months, or you want to have long-lasting system that can stay for months. We can tune the way we want, and we can tune the degradation, we can tune the properties the way we want. Being from engineering side, it's a very fascinating molecule to work with, and we can design materials in different ways. With that, I thank you, and ask any questions if you'd like to have. Open for questions.
Of course, I have to ask from the commercial side. So hyaluronic acid, there are not many approved ones. So the question is yours based on bacterial production or based on animal? Okay. So how do you think would be then the route for approval? Have you been working on the research so that you are able to, prepare the documentation?
We have not gone so far on commercialization side on the gels, but I believe there are certain gels for cosmetic purposes which has been used in patients, and there are many examples. Galderma is producing a lot of hyaluronic acid gels for cosmetic applications. From the medical side, there are applications of arthritis perspective. I don't know if it is approved or not, but there are clinical trials with for arthritis applications.
Yeah, I think it's good to follow FDA and EMA to see where it's going. But thank you for your answer.
Thank you for the nice presentation. I have a question regarding the heparin-based nanoparticles. So, do they have the same efficiency like the exosomes?
Well, exosomes are a black box because we do not know how exosomes work. So it is very interesting that you can deliver drugs using exosomes. But main factor is that exosome also have a sugar coating on the surface and also some protein expression on the surface, which makes them a biologically good transporter for molecules. So we do not know whether they carry heparin with them, whether these exosomes have a heparin coating on the surface because it goes to the brain or to other tissues. We do not know that. But there is a possibility that these sugars are also expressed on the exosomes, which makes them delivering stuff to the brain. But most important challenge with exosomes are that loading drugs inside the system is not that efficient, and the stability of these exosomes are also another question. We can't do long-term storage or transportation. That is another challenge with exosomes. But with these polymer-derived particles, you can freeze dry them as powders, and you can, because they are negatively charged, they're highly stable, you disperse them in a buffer, you can get back the size, which is not normal with any liposomes or any other particle that is commercially available. So these anionic charged particles are highly stable, you can freeze dry them and keep them in fridge for years, and you can use them whenever you want. So that is the advantage, that you have the nanoparticles over exosomes, and it's another factor. So we do not know how exosomes work, but probably there are sugars on the surface like heparin or heparin itself that helps in translocation of the particles to the disease systems.
And do they have the same efficiency in crossing the blood-brain barrier?
I don't think so, because every particle has different, and delivery route is also different. So we have never compared exosomes with our particles, but something good to test. But I don't think they will be same. And even when we did in vivo experiment using IV injection versus subcutaneous, we could see a difference in uptake, and the kinetics and the pharmacokinetics were different. So it will be different system altogether. So you will expect something to be very different from what you get here.
Yeah, thank you so much for the nice presentation. I also have one question. So, like, I'm not expert in nanoparticles. So, but could you please also shed some lights on the toxicity of these particles and the pharmacology of these particles?
Yeah, that's a very interesting question. So why we prefer these glycosamine glycans are because they are polymers produced by our own body. So when we take a synthetic polymer or synthetic molecule into our body, we always have to fight the immune system, because our immune system want to eliminate it. That's the first action it does. It goes to the liver, and most of them ends up in the liver. So when we use these polymers that are produced by our own body, it is natural system. So it has its own biochemical reactions with our blood. We have not presented here, but we have done human blood studies with human patient samples and human blood samples to show how these particles survive and how they regulate the complement, the coagulation cascades. So when we look at the synthetic particles, synthetic drug delivery systems, you always have the immune problem or rejection problem. But when you work with these polymers, which are produced by ourselves, we don't see that problem, that as something very significant. Thank you. Thank you so much for your time.
Our next speaker is Professor Pravat Kumar Mandal, Director in Charge, National Brain Research Center, India. Professor Mandal is a graduate from IIT Madras and Tata Innovation Fellow. He worked as tenure-track assistant professor at the Department of Psychiatry, University of Pittsburgh Medical School, and associate professor at Department of Radiology, Johns Hopkins Medicine, USA. At present, he is working as a scientist in NBRC and honorary professor at the Florey Institute of Neuroscience and Mental Health, Melbourne. Professor Mandal is working on medical imaging and developed novel technology for brain signal processing platform, Kalpana, Indian brain template, Brahma, big data analytics, Bharat, and brain imaging-based database and data analytics, Swadesh. The seminal work of Dr. Mandal is the discovery of brain antioxidant glutathione and its quantitation in Alzheimer's patients and cognitive profile correlation. Now, Professor Mandal is going to share his novel research findings on early diagnostic biomarker for Alzheimer's disease using imaging-based non-invasive platform. Professor Mandal, over to you.
Yeah, so this is our beautiful campus, National Brain Research Center, which is outskirts of Delhi. This is specialized for brain imaging research in India. So here we have various disciplines of brain research, and I'm talking about the neuroimaging component of this one. So this talk today is on Alzheimer early diagnostic marker. This is performed on patients in collaboration of All India Institute of Medical Science, New Delhi. So what we have discovered, as was clearly told, that we have identified a novel early diagnostic biomarker. So basically, there are many things. Imaging-based non-invasive technology was developed, and I will walk you through. So various papers we published from in this center in association with various collaborators around the globe, and one from even Europe. So you can see the researcher from all over. This is not independent work. We need collaborations. And you can see that Bergen University of Norway also part of it, Johns Hopkins and Florey, and at AIMS in New Delhi, and UPMC University of Pittsburgh Medical School, where there is a collaboration is there. So this is a collaborative efforts, but the technology developed here, application here. So MRI spectroscopy is a very powerful technique. Actually, brain biochemistry you can monitor. And it is not only monitoring some decrease or increase, you can actually can get the quantitation of the metaboloids, and also same time you can talk about the disease severity, because we are not the imaging persons. We have a strong base of neuropsychological profiling at our National Brain Research Center. So this is why we correlate brain chemicals, functional behavior, and anatomical structures, as well as the neuropsychological behavior. So it's actually a deadly combinations to get the truth of these molecular processes. So here we can see that a persons people can go to the MRI center, we can find out their brain pH in 13 minutes. Non-invasive, we can find out their how much antioxidant level is there. We also find out the normal metabolites like N-acetyl aspartate. We can find out how much ATP levels, ADP levels are there. So these are all common things we have standardized at National Brain Research Center. Now, in this process, I want to share with you that we have a US patent granted. This methodology, please note that these neurochemicals are very, very small amount. So signal coming from there in a tiny areas of a patient of severe Alzheimer, it's a daunting task. So we have to have a precise technology to collect the data. We have to have a precise mechanism to get this signal processed correctly and then correlate with the disease process. So this has been very heartening that US office has given a patent approval. Now there are several papers we actually. So this is the first paper published in the scientific literature where we actually show that the antioxidant level from Alzheimer patient can be detected and non-invasive manner. So this is why this paper is first time in the literature that has been developed. Yes, this is the another important materials. I was listening to the earlier speaker, the dopamine they can detect, but here we can detect in using MR spectroscopy, the GABA, which is very much profound role in Parkinson disease, epilepsy. We can detect it in a precision manner at any part of the brain. So this MRS technology is very, very useful, and it is also useful in Alzheimer's disease. So you can see the different parts of the brain that GABA has been detected and that has been distributed. This is a technological demonstration that GABA can be detected at various parts of the brain. Next one, please. Yes, this is the first breakthrough paper came out where we can see in published in one of the. Yes, please hold on. I'm just going to explain this one, that different brain regions of the Alzheimer patients, which is in Alzheimer, generally hippocampus and frontal cortexes are affected. So we were able to measure in a quantitative terms, and also the neuropsychological observation has been correlated with the antioxidant level in the glutathione. So this is the breakthrough in this Alzheimer disease research, that antioxidants are correlated in the brain with the neuropsychological behavior. Next one, please. Yes. So this one, you can see that different two interesting observation we found it out. As you know, the memory is a major problem in Alzheimer's disease. The memory goes away in the early stage. So we found it out first time that hippocampus, the glutathione levels depleted significantly when a normal person, healthy person converted to mild cognitive impaired subject. That's the early Alzheimer's disease. But when after some time, when early MCI patient, after 4 or 5 years, they converted to full-blown Alzheimer, the frontal cortices glutathione level significantly decrease. So this is the two distinct approach, two distinct regions has been discovered from the same biological psychiatry paper. So next one, please. So here we are saying that antioxidant is now well accepted that it is depleted. Now we wanted to find out to the distribution from different origin person or different MRI scanner, so that it is not specific to India and one of the centers, it is across the globe. So people all over the world, we actually can see the continent. We use the Norwegian center using the G machine, and we have a Siemens, we have a Philips scanner. We have found it out. No matter what machine you use, you can amount remain there, and there is a pattern among the different people from different continent. I had to travel many places to collect this data. So this is a very important observation that this antioxidant is there, different amount, but it is pattern is same. So next one, please. It took us also 1 year time to collect this data. So what we found it out, there is another important observation. We actually cross-checking our work that anterior cingulate cortex and posterior cingulate cortex. This is the two important regions. There is always active and they are becoming impaired in the case of Alzheimer's disease. The human brain mapping, one of the very good journal, we publish it here, we show them, yes, glutathione levels are significantly depleted in the case of Alzheimer's. So it actually proved. Now there was a very important observation. Cerebellum is another area which is not affected. In the case of Alzheimer's disease, there is no glutathione change in a full-blown Alzheimer's patient and a healthy old. So this is that means selective areas which is actually involved in the Alzheimer's disease process, there is a glutathione levels changes significantly. Next one, please. So here we are seeing it is coming. I can see that pH of the brain of various regions are critical. And as we said, only Alzheimer's disease, the pH become alkaline in the hippocampus. So there are associated phenomena happen in the case of Alzheimer's disease. I have mapped out from the literature what are the cases they found it out in autopsy, but it is extremely difficult to measure the pH for all diseases. So we have able to correctly measure the pH in Alzheimer's patient. Next one will be showing a pattern. Please go to the next one, please. So here we have our target area is the hippocampus. So hippocampus level question comes in, glutathione level antioxidant is going down. So how about their pH? Which one is the early event among the two important factors? What we found it out, when the person turns into MCI, there is not much pH changes to the alkaline region. So the glutathione depletion is the early event, then the pH. pH changes is a second phase observation. So this is why recently we published this work. Basically, we are trying to correlate. There are different events and we are trying to find out which is the primary event for the disease process. Because we at any cost, we have to understand the disease, we have to arrest the disease. Next one, please. So here, what I actually proposed is left hippocampus and right hippocampus, both sides glutathione was measured. This is a raw data. What is very interesting to know that left hippocampus in the case of Alzheimer's disease is affected most profoundly than the right hippocampus. The glutathione level and the pH level also monitored or changes accordingly. So this is why we have identified that left hippocampus is the primary area to look at it before in the clinic, and then we can move on further. We don't need to look at various other regions. So that is a critical message is published in Journal of Alzheimer's Disease. Next one, please. So this is a graphical representation of three different stages, healthy old, mild cognitive and Alzheimer, how the pH changes happen. So basically that's what I explained to you, that is a graphically showing that glutathione changes is the most important event in the case of Alzheimer's disease. Next one. This is just next month. This is another very, very important discovery happened. It came in brain. It is coming in brain communications in next issue or next month issue it will come. It is a multi continent study where we actually looking for, because iron levels is very important in the case of Alzheimer. It's a pro-oxidant. And there are many clinical trials happen involving iron chelators, and many people died. Basically, people did not know why these people, Alzheimer persons, are on the clinical trial. Some of them are dying faster. So what we actually found it out, the antioxidant level glutathione and monitoring the iron level non-invasively in the brain, first time in the populations of a huge number of Alzheimer patient and Parkinson patient, sorry, in healthy control and MCI, we found it out that iron level changes happen at the last stage on MCI to AD. In the initial stage, glutathione changes depleted faster. So this is why this research came in brain communication, accepted in brain communication. Next one, please. There is some data I want to show it to share with you. Next one. Yes. Here you can see three different stages, because this is a live patients. We are measuring their iron levels on the right side, and the left side we are looking at the left hippocampus. As I indicated to you, there is no need for right hippocampus. We need to do only left hippocampus because patient doesn't want to stay longer in the MRI scanner. So these two things simultaneously monitored and plotted, and this is why the observation is. So the patient, the normal person convert to the MCI AD, glutathione level significantly decrease, as well as the iron level increases significantly. So there is a very nice diagrammatic presentation. Next one, please. Here it is all things has been shown, the glutathione levels, and then to get it accepted in the clinics, the receiver of our ROC curve was generated, and sensitivity and specificity for clinical environment doctors will be accepting this result when it is above 75%. So this is why you can see that a normal person, MCI and AD, that glutathione levels has gone down. Iron level changes at the end of the process, MCI to AD. That explains that why the chelators was not accepting, because it created a havoc for this patient. So some of them are died actually. So this is a work of 2.5 years with a collaboration with a Florida Institute of Neuroscience. So that's what is a 19 authors paper which came in this brain communication. Next one, please. Yes. So we actually modeled it, that why this work is happening. This is a breakthrough work came out that glutathione is actually protecting how this Alzheimer disease is happening. When there is no protection, one of the residues called methionine 35, which is actually structural changes happen, and actually they actually initiated the oligomerization of amyloid beta peptide. So you can see that one, when a good system, very good protection, there is no problem. Amyloid beta peptide remain monomer, but in the right side, in a pathological consider, there is a oligomer formations, and actually is just published last month in ACS Omega 2022. That mechanism has been given, and the data, actually I presented this one in brain communication, that raw data. This actually motivate us to give that model for this one. So this one I just want to screen, as we saying that huge amount of data has been collected. So Swadeshi, as was introduction time, it was mentioned, all data are going to be shared throughout the world. We have huge number of patients data. These brain chemical data are available at National Brain Research Center. We will be sharing. So this database has been generated, and all the data analytics. So nobody has to go other places to use the softwares. Data analytics, data, all are in one place. So people has to log in. They can use our servers. They can get it processed in free, and they can take their data. We don't need anything from anyone. So this is we want to use. It is a government of India supported in a very interesting. project. So that's called SWADESH, so that we want people throughout the world should be able to use this one. Next one, please. So this was announced by the Government of India Press Information Bureau that this has been a nice novel database has been generated, and this is open coming up for using. So this data is unique in the sense it is not a MRI data, it is a neurochemical data, and nowhere still now available this kind of the changes, glutathione, GABA, glutamate, glutamine, nowhere it is available. So we'll be able to share this one to everyone. Next one, please. So we have developed a artificial intelligence based analysis, early prediction. So this is why we have published this work in ACM Computing Survey, one of the top journal in computer science, that how these features will be utilized to predict the diseases. So that's what we have developed. We have a patent in our another patent under review called Gauri. So that will be able to do this one. This is the initial paper we published recently. So the ACM Computing. Next one, please. So this one, what we actually try to say, how the AI-based approach algorithms will work, and that's what actually showing that these various features. It is not MRI-based features. All are working together so that decision-making process is precise. So this will be various type of features will come from various modalities, and that will be combining together. It is very much in testing stage, so that soon it will be coming out for the hospital use. So this is why AD hypothesis we are actually revisiting. That's saying that oligomer formation, amyloid beta peptide has to be rethinking. So what is oxidative stress is an important observation that need thorough clinical trial. So that's what I was saying, this article, because this has to be there. Next
Professor
one.
Mandal,
Yes.
can you please wrap up? We are going over the time.
Okay, last one I can just go through. Last one, yes. Okay, so just I see that. So in conclusion, we first time detect the glutathione is available, and this monitoring is the critical, and iron level has to be monitored at the last stage. So this is the two important observation, and I want to share the thank you slide, last one, that is very few seconds it will take. Okay, so these are the different collaborators and others. So they actually contribute, and we are working as a team. Thank you very much. I'm sorry for the technical glitch we could not control. I took about 10 minutes extra. Thank you very much. Thank you. I'm open for any questions.
Yeah, thank you. We thank Professor Mandal, and let's move on. Our next speaker is Professor Jari Hyttinen from Tampere University, Finland. Professor Hyttinen is a full professor and head of Biomeditech unit at Faculty of Medicine and Health Technology at Tampere University. He received his master's and PhD from Tampere University of Technology. He has been visiting researcher in University of Pennsylvania, University of Tasmania, and Duke University. He has acted as a visiting professor at University of Wollongong, Australia, and ETH Zurich, Switzerland. He has served in many positions of trust and graduated over 110 masters and 18 PhD students. He is a co-author of more than 390 scientific refereed articles and some patents. He is also co-founder of the company Inject. His laboratory develops novel computer simulations on cellular biophysics, body-on-chip technologies, and in vitro 3D imaging methods for future personalized medicine. Now, Professor Hyttinen is going to share his vision on future personalized medicine through integration of biomedical and engineering sciences. Professor Hyttinen, the floor is yours.
Okay, thank you for a very nice introduction, and nice to be here, and thank you for invitation. I think what we heard in the last couple of talks, I think they were already quite nice introduction to my topic, because you could see that there's really integration, a lot of things to achieve these new knowledge of diseases. And I'm coming from Tampere University, and my kind of idea today is first of all tell you what we have done in Tampere on to achieve this kind of integration, and give small let's say glimpse of things that what we are doing on this topic as well on science side. But mostly this is really telling what we are doing in Tampere in like organization level. Okay, this is just kind of introduction. We heard about brain, but same thing happens in kind of heart as well. If you think about it, there's a lot of disease. We kind of see people dying, for example, in cardiac disease, like these young athletes. We all heard about these news. And of course, this is a disease where there's a genetic kind of problem that causes this disease eventually. But equally, there's a phenotype of these people who will actually die, who is not dying, why certain genetic kind of problems are causing death, sometimes not. So it's really combination of phenotype and genotype. But if you are only genotyping, it is not enough. So we need to have both kind of doing well. And so for phenotyping, we actually need better methods. Like previous talk, there was new imaging methods appearing where we can much better view what is happening in the brain. That was really phenotyping of these patients. Okay. And so this is my view, and I think many others are agreeing this, that if you think about future trends in medicine to really achieve this kind of, let's say, precision medicine and better healthcare, the way we can do it is only integration of many areas. There's kind of traditional biomedical engineering areas like imaging, sensors, wearable things, then kind of using AI on mobile phone use or genetic information. But there's a lot of emerging areas as well, from molecules to cell models and others. And really to achieve this, there's kind of challenge how we can facilitate this integration. Collaboration is of course one way, but of course what we could do also in kind of our faculties or universities. And at Tampere, or in Tampere, we had actually quite nice opportunity a couple of years ago because we had two universities, Tampere University of Technology and Tampere University, and these were merged 4 years ago. So we had the chance to do something new, possibly. And what we decided to do is build Faculty of Medicine and Health Technology. And it's bringing engineers from our TU Tampere and biologists from Tampere University and clinical medicine from Tampere University and close collaboration with Tampere University Hospital. So this is our attempt to kind of see the future, try to integrate these people. Of course, we have a lot of things to do in the future, but this is kind of the starting point of hopefully something that we can really achieve. Also better integration of these disciplines. And of course, one area is education. And so far we have little bit integration. There's things to do still. We have a combined program of biotechnology and biomedical engineering with a lot of actually main topics. And we heard about the exciting work of Professor Omen as well. That is part of the biomaterials and tissue engineering program. But there's also nano and microtechnology, health technology, and informatics, and also bioinformatics, cell technologies. And we have also, of course, MD program. We have medical school under the same roof. And we have integrated the PhD program together. And of course, one interesting or important area is to how we bring our innovation and research to, let's say, outside world, how we impact the society. And one way is, of course, commercialize these. And for that, we have made actually quite large package for really study module for commercialization of medical devices and software. And we are part of also activities, translation these ideas. And one interesting program, we are kind of one of the leading in Finland on that, and that is the Spark program that is actually started from Stanford, and it's brought around the Europe and the world. At the moment, I don't see any places in India on this program, but maybe that could be one way of collaboration as well. And what we have also, we have translational research centers that are more like concentrating on certain clinical questions. There's also more like advanced technology center That is the last on this list. But these are the ways where we can, let's say, use our wide expertise and concentrate on certain clinical questions. And like whether it's cardiology, celiac disease, cancer, for example, and really integrate our research. And of course, through this integration of engineering and biology, we have also been able to build quite nice laboratories are quite, let's say, I would like broad in sense of what we can do. We can build devices from nano devices, micro devices. We have different kind of cell models, cell culturing system. We have a system where we can actually produce cells and tissues for implantation of humans. We have various bioimaging systems we build ourselves as well. You heard story of biomaterials. One story of those. We have quite a lot of facilities for those as well. And also, of course, more traditional ones like how to measure human or how to image human. So that's about the kind of, let's say, more like administration what we do. But there's one example I would like to tell, and that is the where I belong as well. So of course that's why I bring it here. But it's also a nice example of how we integrate people, and we have done it, of course, longer than actually the new faculty. We have collaborated long time, but we have one of the centers, Academy of Finland of Excellence, and this is body on chip center of excellence, a body on chip technology. And we have 6 research groups, something like 60 researchers working in the center, financed by the academy. And our idea is, of course, to body on chip system. Body on chip is what it means is that we are trying to build human body on test tube. And they're also called nowadays a microphysiological system. That is what Americans would like to tell them. So what these are, this is one example from Professor Pasi Kallio's microfluidic chip in the center, but that's part of that story. So first we need people who are delivering the cells, giving the cells on and fabricating new tissues for it. And not just cells, we are going really for tissues, and that is, I'll touch that later. And then we need ways to control the tissues and cells on those chips, and that is microfluidic system, other control system. We need technologies for interrogation, imaging, sensors, ways to see what the cells are doing there. So if we have the cells, we can put drugs. We can see what the drugs are doing by imaging, by sensors and others. And of course, we need the biomaterials that Professor Oomen was already telling here. A lot of hydrogels actually is now there. Also some products from the company. And what I see, I'm working on these boxes that are framed. So on imaging, another side here is computational modeling. And computational modeling is kind of what we heard already on this, how we digitize biology. If we have this system functioning, how we can understand deeper what is going on there, whether it's AI, but also making the biology digital and making models of those cells there so we can understand their behavior better, or simulate them in computers so that these systems are quite, let's say, still cumbersome to use. We cannot have hundreds of thousands working together or parallel. So in computers we can do a lot of more, and then go back for the bench and see how a prediction in computer work here on this system. These are just 2 examples I've selected, just more. This is very old slide, but just kind of shows what can be done. This is one of the first times, more than 10 years ago, we already had human cells beating on these chips that are called microelectroarrays. So they are like your ECG electrodes, but they are on bottom of the test tube there, and you can see the heart beating, and there you can see the signals like ECG coming from the heart cells. And then we can add drugs, and we can see how the cells react. Of course, and nowadays these cells can be your cells. So we can have your cells turn to stem cells and turn to cardiomyocytes, and we can test how the drugs are functioning there. And of course, what we can do a lot of kind of different tricks here. We can change the environment. We can change the oxygen content. We can have ischemia of the cell or doing something else there and see how these combined system work. And of course, they can be different genetic background of people and we can have disease on this. The disease on genetic disease, or it can be disease gained by the, let's see, like oxygen deprivation, ischemia, for example. But of course, what we can do as well, this is one example of, we can actually replicate the function of cardiomyocyte in computer, have quite elaborate system to have full functional electrophysiology of the cell, also biomechanics of the cell, beating cells in computer, and then generate kind of population of these tens of thousands of cells beating in computer. If you could think there's 10,000 people or 10,000 different kind of cells of the heart, we can have diseases in computer cells. And then we can test drugs, because many drugs are actually affecting these same kind of system that we have built components. There are ion channels, exchangers, and if you change the parameters, that is the effect of drugs. I don't go into detail what we have done here, but you can see that we can have distribution of the effect. It's just like having population, and we can have diseased cells, healthy cells, and like here we show that when you have really diseased cell, not just the genetic disease, but cells that are behaving like disease cells, the drug effect is better on those cells than the cells that are normal, or the cells that are actually quite normal despite they have the genetic problem. And so we have done actually quite a lot of work. Let's see whether I can. Where's my mouse here? Sorry. So we have done actually quite a lot of work on this and quite a number of publications over the years. And we have shown that this can be done. We can do it on dish on the lab. We can do it in computer. We can have these human-derived stem cells and cardiomyocytes, neural cells, other cell types, and we can model the drug effects. We can model the inherited diseases, and we can have injury like the ischemia or other diseases. But of course, from cells to human, it's actually quite a jump. So of course, we would like to do is kind of bridge this gap from these cells to human scale. And of course, there are multiple ways to do it. And one way is that we can build computer models of each of these levels and try to migrate the information from one level to another. And that's what we aim to do as well. Modeling cardiac system in heart level or modeling in one single cell. Actually, that colorful picture on the back is from my own PhD thesis that was a couple of years ago. But just to kind of show that there's a kind of tradition on this direction as well. There's actually video here as well. You can see mouse heart, and it's imaged from micro-CT how the structure is important in heart as well. But in order to do this, there's another direction, and that's actually the building in dish better models, not just having cells, but building tissues. If you think about your brain, we heard about the story of the blood-brain barrier. You heard a story about different cell types in the brain. So your brain is actually mostly something else than neurons. There's a lot of other cells, and these other cells can start behaving pathologically. So in order to have it on dish, we have to actually combine these systems in dish as well and build these body on systems with multiple tissue cell types. And at the currently we have like a combining vasculature and nerve cells, having heart cells and neurons, because in your heart there's a lot of neurons as well, they're interacting, and vasculature and adipose cells, the fat cells, to study what is happening there in metabolism in the body. And of course, we hope that in a couple of years, we are having a little bit more complex system, even having multiple of these components together, and we can build certain level of simulation of human body, looking into interaction in diseases, especially metabolic disease, but also interaction in like metabolites of the drugs. And it's really multiple levels of engineering, multiple levels of biology integrated on these systems. So I hope I convinced that Tampere is a nice place to come for science. And if you are looking for collaboration in commercialization, we are happy to be partner of that as well. And of course, there's a lot of more happening. You heard Professor Orman's story. There's something more coming from Tampere, not our faculty, but just showing there's other people there as well. And of course, we have almost 100 research groups in faculty, so there's a lot of things to happen. And if somebody is interested in more about computing cardiology, come to Tampere in a couple of weeks. We'll have 350 world leaders on this field. Thank you.
Thank you, Professor, for very nice presentation. I just have one question. Few years back, one of my friend's son died. He was just 18 years old, and he's just going to start his medical sciences studies in Sweden. And he have sudden cardiac arrest, and he passed away. So, is there any way we can have some kind of system to examine all the kids at young age of these kind of diseases?
Yeah, I think in some countries, for example, was he athlete or yeah, there's really on young athletes there's some screening programs. Especially I think in Italy is in heading this. Of course, the screening start from the gene, so that can be done. But then whether that there could be a gene that kind of raising the chances of having sudden cardiac death, but still it doesn't need to say that there is a sudden cardiac death coming in for this individual, and that's the problem, and that's why we need the other models. I know that we have like in our labs with Professor Katrina Aaltosetä, he's really interested on this, and having the actually cells of these people in the lab and seeing why some have really high risk of these arrhythmias and sudden cardiac death and others not. And actually we are I'm just coordinating one application to EU where we try to put everything together and have a lot of data and AI as well mixed together. The deadline for application is at the same time as this meeting, so that's a little bit busy time for me.
And unfortunately, Professor Subra Chakravarti is unable to attend the event due to some personal emergency. And yeah, that is the end of the plenary session 1. And it is really great to see a lot of interesting talks and stimulating discussions related to the topics from biomedical engineering to nanomedicine and brain research, which is the need of the hour. I would like to thank all the speakers for their great presentations and stimulating discussions once again.